Welcome back, everyone. Our second speaker is Dr. Bochra Bahri from the Department of Plant Pathology at the University of Georgia. She conducts research in the area of turfgrass and forage pathology to include aspects such as pathogen biology, population genetics, and host resistance. Bahri grew up in Tunisia and received her engineering degree from the National Agronomic Institute of Tunisia. And she also obtained her master's and PhD degrees in 2 universities in France. She has held postdoctoral positions in molecular biology at the University of Aarhus and in genomics, genetic analysis, and mapping at UGA. Welcome, Bochra. The floor is yours.
So much, Bonnie. Can you hear me well?
Sure can.
All right, perfect. How about the screen? You see well my presentation?
Yeah, looking great.
All right, perfect. Thank you so much. I'm very happy to be with you this afternoon. We had a very great session with Alison, very intense. I hope you're not too tired. It's going to be also, we are going to be together for 1 hour. Let's get started. I'm going to talk about fungicides today. So what I want to present you today, and I want you to have, by the end of this talk, I want you to get knowledge on different aspects of fungicides. And this is what I'm going to talk to you about today. First of all, what is a fungicide? Trade name, generic name, chemical names. What are the different types of fungicide formulation that you can have? What are the different mode of actions of fungicides? The chemical classification of the fungicides, and what is the fungicide resistant problem? And this is also a very, very important part of my talk today. So what is a fungicide? A fungicide is any chemical that will kill a fungus. So it's basically, the fungicide will have a fungistatic effect that will inhibit the fungus, and it can be at any stage of the cycle of the fungus, from the spores, the mycelium, etc., or the reproductive structure. All fungicides that you know are characterized into, they have 3 names. The first one is the trade name, and the trade name is given by the manufacturer. The generic and common name, this name is basically an abbreviation of the active ingredient, and the chemical name, which is the full complete name of the active ingredient. I'm giving you a few examples here. One basic one, for example, the thiophonate methyl. This is the generic name. The trade name will be, for example, Krylis 3336. And the chemical name, most of the time, the chemical name is a scientific name, actually, and it's going to be more complex. When you see a very complex name, this is the chemical name of the active ingredient. Why is it important to know the generic or the common name? It's important because for the fungicides, most of the time, the nomenclature is going to be very confusing. Example here, you will have several trade names that are very, very close to each other, but they are basically different active ingredients. So it's very important to know the generic name of the product and not the trade name of the product. Another point also is sometimes the trade name, you will have a separate trade name for the same active ingredient. The thiophonate methyl, for example, will have more than 19 trade names. One that you probably are using a lot, for example, the trade name for azoxystrobin. We also have several trade names, Bond, Quadris, Heritage, etc. You can also have several companies that produce the same product. Propiconazole is produced by several manufacturers. And of course, what differentiates these products is going to be also the price, because the price differs between the different producers. One important thing that you need to look at is the fungicide labels. In the fungicide labels, you will find the entire information about the common name. Most of the time, it's going to be in the active ingredient name. You will see the common name here and the chemical name, which is a more complex, more scientific name of the product. And you will see in the front the trade name. So all this information you can find. And what is also important about the fungicide label is by law, certain kinds of information must appear in the fungicide label. And it's your responsibility to read the fungicide label and understand the direction in the fungicide label. And you will see a lot of information in the fungicide label. For example, anything that is related to the environment, to the hazard to human and in animals. When you can use the product, the agricultural use of the product, the requirements, how you can store and dispose of the product, how you can manage the use of the products and reduce the emergence of the resistance. You can also find information about tank mix compatibility, what you can mix with, etc. So it's very complete information and very useful information. So now I'm going to talk about the fungicide formulation. So fungicide formulation, Alison already talked a lot about it. I'm not going to go into detail because she already covered a lot of it. So when we talk about fungicide, you have two things in the fungicide: the active ingredients plus inert ingredients. The active ingredients is the one that is going to basically affect your target. The inert ingredients does not have any fungicide activity. It's only here to enhance the effectiveness of the active ingredients. And there are several inert ingredients: carrier, antifoaming, weighting, sticking agent, etc. And the way how the active ingredients and the inert ingredients are put together is called fungicide formulation. And you can have different fungicide formulation. I give you an example here for granule and powder. Alison already went through all these different fungicide formulation, from water immiscible liquid, water soluble powder, wettable powder, flowable, dust, granule, etc. And you have different abbreviation that is also in the fungicide label that you will find it for each of the different formulation. When you look at the fungicide label, so you will see next to the trade name the formulation of the product. For example, here is a flowable, okay? Why is it important, the formulation? As I say, the inert ingredient will help to increase the efficiency of the active ingredient. This is one example that I'm going to show you. For example, this is fungicide, two different fungicide, Bayleton and Armada, that were used in different formulation, either suspension concentrate or granules or wettable powder, against brown patch. And this was done on tall fescue. And you can see when you compare the effect of the fungicide to control the disease, and you have the non-controlled one here where the disease has a very high level, you see that the use of the fungicide will reduce the disease, but it also, different formulation will have an impact on the disease. For example, here you can see the granule. In the granule, you see still more disease than the friable suspension concentrate, etc. So really the way how it's formulated, you can have an impact of how efficient the product is going to be on the particular disease. And this is just a particular case for brown patch on tall fescue. For some other disease, the granule could be better to use than wettable powder, for example. All right, so now I'm going to talk about one of the most bigger section in my talk is the activity, mode of action, and chemical classes of the fungicide. The fungicide can be grouped according to 3 axes: topical activity, their mode of actions, and the chemical structure. I'm going to talk first about the first one, the topical activity. So topical activity, we have two topical activity. The fungicide can be either systemic or a contact fungicide. Contact fungicide will only act on the surface of the plant and will inhibit the development of the fungus on the plant. It will not penetrate the inside of the plant, and it will not inhibit the fungus that is already inside the plant. So, for example, here is the surface of the leaf, and the fungicide will stay and will redistribute in the surface of the leaf. And this kind of fungicide, as I said, do not penetrate the plant and will only target the fungus on the top of the leaf, and mostly it's going to be spores that are on top of the leaf or mycelium of the fungus that are still on the top of the leaf. Contact fungicide are also called non-systemic or protection fungicide. So as I say, this kind of fungicide, when you spray it, they will only be efficacious on the pathogen that is outside the plant, on the surface of the plant. They will not penetrate inside the plant. So everything that is not sprayed, the leaf that is a part of the plant that is not sprayed, or every new growth will not be protected by the contact fungicide. The other category of the fungicide is the systemic fungicide. Systemic fungicide are fungicides that can be absorbed inside the plant, and they can move to the roots, to the leaves, etc. And this kind of fungicide can inhibit the fungus both ways, on the surface of the plant and also inside the plant, in any part of the plant, the roots, the leaves, the stolon, etc. Inside the systemic fungicide, we have different categories of fungicides. It depends on the level the fungicide is able to penetrate inside the plant. Some fungicides are localized, or we call them local systemic. These fungicides basically penetrate inside the leaf, but will not diffuse inside the plant. We will stay around the site of application. The translaminar systemic fungicide are fungicides that will penetrate inside the plant and will go on the opposite side of the leaf. So it will protect from one side to the other side of the leaf. Xylem or acropetal fungicide are fungicides that will penetrate inside the plant and they will move towards the tip of the leaf. So basically, wherever you apply the fungicide, it will go towards the top of the leaves. The phloem systemic fungicide are fungicides that will go inside the plant, but they will go towards the base of the leaf. So wherever you apply the fungicide, will penetrate inside the plant and will move towards the base of the leaf and go inside the plant, and it can protect any stage, any level of the plant. The amimobile fungicide are fungicides that do both move towards the tips or move towards the base of the leaf. An example here, so translaminate fungicide, as I told you, they will protect everywhere we're spraying the plant, they will protect the plant on the contact, and on the opposite side, the fungicide will penetrate inside the plant, but only move towards the opposite side of the leaf. So it will only protect the leaf that you spray. It will not protect any leaves that are not sprayed, and it will not protect any leaf, any new growth leaves. However, when we talk about Xylem systemic fungicide, this kind of fungicide, because they move inside the plant, they would protect the leaves that were sprayed, protect also the leaves that were not sprayed, and any new growth. So these have a much bigger ability than any other fungicide, systemic fungicide that we talk about. One important thing to know is how we use and when do we use this systemic fungicide, and when do we use this contact fungicide? Because of the ability to only inhibit the fungus on the surface of the leaf, the contact fungicide are only efficacious before the infection. So when I say before the infection, is before you see the symptoms on the plant. However, when we are talking about systemic fungicide, you can use them either as a preventive application, so before you see any symptom, before the infection, or you can use them as curative method of control after you see the symptoms or during the incubation period of the pathogen. The incubation period of the fungus is basically the time that is between the infection, when the spore germinate and penetrate inside the leaf or any organ of the plant, and the symptoms that you see on the plant. And during this time, of course, you don't see any symptoms, so you don't know if the plant is infected. But during this time, only the systemic fungicide would work. The contact fungicide would not work. Okay, so contact fungicide are mostly done as preventive fungicide. Systemic fungicide will be efficient either as preventive or curative control of the disease. Just to show you here, this is dollar spot on turf grass. You see that this is the control, untreated control. This is a preventive fungicide application and curative fungicide application. You see that the preventive with the systemic is really very efficient, even more efficient than the curative fungicide. So it's most of the time it's better to prevent the disease and do an amplification before that the pathogen is here than doing a curative, trying to do a curative application and try to reduce the disease. The fungicide can also be divided into mode of actions, and there is two types of mode of actions. Some fungicides are going to be site-specific inhibitor. Some other fungicides are going to be multi-site inhibitor. Site-specific inhibitor are fungicides that will target one pathway or one particular enzyme in the fungus. An example here of enzyme, for example, dimethylase for the triazole, ubiquinol oxidase for carboxymal. And these site-specific inhibitors are highly specific, and you will have a very high chance and increase of the fungus to become resistant to this kind of fungicide. However, the multi-site inhibitor, they will target different sites inside the fungus cell, and you will have less likely to have resistance against this multi-site inhibitor fungicide. So as some example here of site-specific fungicide, the DMI fungicide, for example, will inhibit the stereosynthesis in the membrane, so it will target the membrane. The benzimidazole will target, for example, thiophanate methyl will inhibit the DNA synthesis, so will target the nuclei. SDHI fungicide will inhibit the respiration complex, so the target is the mitochondrium here in the cell, and it's the same for the strobilurin, but it will target another complex of respiration in the mitochondrium. So strobilurin will target the complex 3, and the SDHI will target the complex 2. The multi-site fungicide will have different targets, many targets in the fungal cell. So it will target cell membrane, the vacuole, the nuclei, the mitochondria, the ribosome, etc. And they are less likely to induce resistance in the field. All right. So the Fungicide Resistant Action Committee, or FRAC, had developed a code number to basically classify the fungicides according to their mode of action. So all the fungicides that have the same mode of action are grouped together in the same group. And when you look at the fungicide level, you will find this information. And usually it's up above the trade name, you will see the fungicide group or FRAC mode of action or FRAC code that is used for each fungicide. And with this mode of actions, we can classify the different fungicide into several chemical groups. And when we talk about fungicide, there is over more than 14 different chemical groups. I'm going to present today the most important one, the one that you use more frequently. The main chemical groups in the site-specific fungicides. The first one that I'm talking about is the DMIs, demethylation inhibitor. So the classical chemical class is DMI. There are many fungicides that are included in this class, and many generic names like propiconazole, for example, triadiméphon. All of them are going to be systemic, so they penetrate the plant. These demethylation inhibitor were introduced in 1970s. The FRAC code is number 3. As I say, these DMIs will target the sterol synthesis inhibitor. They'll target the sterol. Here is a compound that is in the cell membrane of the fungus, and it will inhibit the production of this ergosterol. These DMI are really widely used. It's a very large class of fungicide. It's very effective on all water mold like pythium, etc. As I say, it's systemic, and they have a moderate risk of resistance. Several examples here: triazole, propiconazole, etc. Another class of site-specific fungicides is the strobilurins. Strobilurins, here, you can find several that are localized penetrant, so they only penetrate and stay in the point of inoculation, or systemic. For example, the azoxystrobin here is a systemic fungicide that belongs to the strobilurin. So the strobilurin were introduced in 1990, FRAC code number 11. They basically target one specific gene, the cytochrome BC1, in the respiration 10. It's a wide spectrum fungicide. It is effective against oomycete, ascomycete, basidiomycete, etc. But it has a very high problem for resistance that was observed in this fungicide, mostly for azoxystrobin, for example. Another class of site-specific fungicide is the benzimidazole. So the benzimidazole are going to be systemic fungicide. The most common one, trade name is the tufenate methyl, and there is a lot of, as I said, there is a lot of trade names for the same generic name. They have been introduced in 1970. The FRAC code number is number 1. They target one specific gene that is called the beta-tubulin gene. This beta-tubulin gene helps the cell division, help to create these microtubules during the mitotic phase, during specific mitotic phase during the cell division, and basically the cell cannot divide anymore. So the fungus cannot divide anymore. There is no cell division anymore. These are active against ascomycete, but they are not active against oomycete. And here also, we have a very high resistant problem against this benzimidazole. Another class of site-specific fungicide, the carboxamide, and here I give you 2 examples of systemic carboxamide. They are also called SDHI, succinate deshydrogenase inhibitors. 2 examples of trade names, Prostar and Emerald. They have been introduced in 1970. The FRAC code is number 7. They also target the respiration, but a different complex as the complex number 2, different complex and different in the mitochondrium. The older SDHI were mostly specific to basidiomycetes. However, the new one have a much broader spectrum and will be effective against much more classes of fungi. And some resistance has been reported for this class of fungicides, and it's classified as medium to high risk of resistance. And here I give you some example of the different generation of SDHIs that were produced. At first generation, carboxine, second generation, boscalate, for example, and third generation, you have other fungicides that were produced. And these are the newest ones that have a broad spectrum. Another site-specific fungicide, carboxymide. So the carboxymide are locally penetrant, so they stay in the point of inoculation, basically, application. Two imported examples, Iprodium and vinclozolin. They have been introduced in 1970. The FRAC code is number 2. They have a broad spectrum, but they're not efficacious against powdery mildews. And they have a high to moderate risk of resistance. And it's mostly, there are several cases of botrytis resistant to dicarboxymide that were observed. And the two examples that I give you, Iprodium will inhibit spore germination and mycelium growth against Sclerotinia, Monilia, or botrytis, for example, and vinclozolin, again, is also efficacious against Monilia, Sclerotinia, etc. Other class of site-specific fungicide, phenylalanine, aliphatic, and fosfanate. They're also very important, and as you can see here, they belong to different FRAC codes. They're all systemic, and they target different sites. For the phenylamine, they target the RNA polymerase 1. Aliphatic, they target the cell membrane. For the fosfanate, is still the research are still not sure what is the exact target of this fungicide. All right. So now I'm going to talk about the multi-site fungicide. So multi-site fungicide, you also have several examples that you need to know. There is some fungicides that are classified into the aromatic hydrocarbon class. For example, chloronep, etc. And these are going to be contact fungicide. You also have the dithiocarbamat, example, mancozeb, tiram. They're also going to be contact fungicide. Remember, contact fungicide are fungicides that are mostly efficacious as a preventive fungicide. The dithiocarbamat was introduced in 1930-1940s, FRAC code number M3. They have a broad spectrum. They're not systemic, so they're really contact fungicide, broad spectrum of efficacy. And you have, I give you here, you have several examples that you can use, several active ingredients that you can use, and they are very efficacious against pathogens, but at a preventive application. Other multi-site fungicide, the nitrils and phenylpyrols, they're also contact fungicide. So you see here that most of the multi-site fungicides are going to be contact fungicide. Example here, the chloronitrils, FRAC number code M5, they have a broad spectrum. Broad spectrum, it means that it is efficacious against large diseases of type of fungus. Bravo, for example, here is a chlorothalonil. It's an excellent disease control. They have a very broad spectrum activity and can be used in tank mix with other narrow spectrum fungicide, all the site-specific fungicide that I gave you. This one can be tank mixed with it. It's an important fungicide component of fungicide resistant management. So this is one of the ones that you need to think about. All right, so my last part of the talk is going to be about fungicide resistance. So what is this fungicide resistance and how we can manage this fungicide resistance in the field? So the fungicide resistance is when you see a decrease in the fungicide sensitivity within a population of fungi after repeated exposure of a fungicide or a group of chemically related fungicide. So how does it work? And how do we reach this disease resistance? How do we develop this disease resistance? So when we start here by a fungus population, you can see all the small dots, blue dots here are all the individuals that are in the population and that are sensitive to the fungicide. However, you can see here that in this fungus population, you can have some individuals in red. These individuals are individuals that are resistant to the fungicide. How does this resistance appear in the population? The resistance appears spontaneously in the population, mutation by mutation. And when you have a mutation in the gene, the target gene of the fungicide, you will have individuals that will become resistant. And the mutation appears spontaneously in all organisms, and here in the fungal population. However, is the way how you select for these individuals in your population that you will have an increase of resistance and the development of resistance in the population. So every time when you spray, you will reduce the pathogen population. However, the spray will only be efficacious against the individuals that doesn't carry the mutation, that are not resistant. So it will reduce only the individuals, the population that are blue here, okay? And every time you apply, you will reduce. So from this population, the population will grow. So you will have more and more individuals that carry the mutation in your initial population. So the proportion of the individuals that are resistant will increase over time. Every time you use the same group of mode of action or related mode of action. And little by little, you will increase the individuals that carry the mutation, and you will end up with a majority of individuals that are resistant compared to individuals that are not resistant. And this is how you build your resistance. So the spray that you apply in the pathogen population will create a selection pressure that will, over time, with the repeated application, that will result in an emergence of a resistance in your resistant individual in your population. When you look at the history of the fungicide resistant development in the fungi, you can see that this problem is mainly on site-specific fungicides. So there are few cases of fungicide resistance that were reported before 1970. However, it's with the introduction of site-specific fungicide in the 1970 that we have much more reported problems of resistance to fungicides. And nowadays, this reports of emergent fungicide resistance is much more frequent and is increasing for all different fungicides. And of course, it's only specific to the single site, mostly single site fungicide. There are some cases for multi-site fungicide. So as you can see here, the multi-site fungicide, for example, this one can have been used for over 40 years before the resistance show up in the field. However, if we talk about single site resistance, it needs only, for example, in this case, 2 years of application, and you will have a problem of fungicide resistance in your field. How this mechanism of resistance appears? The most common mechanism of resistance is the alteration of the target site in the fungus. So, if you're for the single site fungicide, this single site fungicide will target one specific gene in the fungus, and if this gene you have a mutation is the gene, you can have problem of fungicide resistance. I give you here several examples of fungicide resistance that appeared. You can see that for the different categories of fungicide classes, the benzimidazole, the strobilurin, the DMI, and the SDHI, the target is always different. The gene targeted by the fungicide is always different gene. Beta-tubulin gene here, a cytochrome B gene, 51, SDH gene. And you can see here there are several mutations that have been reported in that gene, a different position in the gene. And every time you can have a different mutation that appears that give the fungus ability to resist to the applied fungicide. There are several factors associated with the risk of fungicide resistance. And here I'm going to give you some of the factors that are important to keep in mind. Some of these factors are related to the fungicide by itself. As I show you, when you look at the mode of action, the single targeted sites have a very high risk compared to multi-site targeted fungicide. You also have the fungicide group, the history of resistance in your field, the use pattern, how much you're using the fungicide, the application, the application interval, the number of application, and the rate of application that you're doing in your field. So all this will give you an idea of what risk do you have to have an emergence of fungicide resistance. Other factors are related to the disease by itself or the pathogen. Some pathogens are much likely to generate more rapidly mutations in the genome. And this is, as I say, related to the pathogen and its mode of reproduction. The pathogen that have sexual stage and shorter generation time will have more higher risk to have a mutation that occurs or new individuals that appears in the pathogen population. The size of the population is important. When you have a very high infestation, it means that you have a population that has a very, very big size. The more the population is big, the more frequent you will have mutant in your pathogen population. Dispersal mechanism, the number of cycle, if it's a polycyclic disease, a monocyclic disease, a disease that is polycyclic that do several generations in one season during the springtime, you will have a higher chance to have a fungicide resistance than compared to a monocyclic disease that only have one generation during the season. And many other factors will also are related to the environment and the mutation rate of the pathogen and how the environment can impact the mutation rate of the environment, of the fungus, sorry. Other aspects, the aspects that are related to the agronomic trait. If we're talking about annual versus perennial crop, the perennial will have a greater risk because the plant is there. You can have several generations during the year. The host resistance, also very susceptible to infection. The plants that are very susceptible to infection will have a much greater risk of emergence of this resistance, of the fungicide resistance. Crop rotation. So if you do the same cropping, you will have greater risk. If you do more crop rotation, you will have less risk of this fungicide resistance. Availability and effectiveness of non-chemical disease control. All this will lower the risk to have a fungicide resistance in your field. Use of biological control, for example, cultural control. So reducing the fungicide application will reduce your risk of having an emergence, resistance emergence in your pathogen population. How do we clearly detect the fungicide resistance? There are three ways. Of course, the first thing is to observe in the field and see if you have a failure of the fungicide application. So you do your application, you still have a problem of the disease. This is the first way how to basically observe that you can have a case of fungicide resistance. The second way of identify the fungicide resistant strain is to run some tests in the lab. So we can do some sampling in your area, or you can send us the samples, and we run the test in the lab to see if you clearly have a strain that is sensitive or is resistant to several fungicides. The Third way to do it is to also run field or greenhouse trial. So we test it in the field. We inoculate in the field your strain, and we see if it's really with the application of the fungicide, you still have a problem of the disease. I'm giving you just a few examples here that were run in my lab. So a lab test, for example, this is a lab test that was done for 79 isolates of Clarividia that cause dollar spot on turf grass. And these 79 isolates were isolated from the state of Georgia in different counties. And within these 79 isolate, most of them, we found a lot that were sensitive. So all the one here were sensitive to the fungicide, and all the one here were resistant to the fungicide. And the fungicide that we use here is a propiconazole. And you can see that most of the isolates, more than half of the isolate that we have were resistant to propiconazole. And you can see here the pathogen growing on artificial media amended with the fungicide, and some of them, even at high dose of fungicide, they still can grow in vitro tests. In the field, the same, we test several fungicides, Heritage, Spectro, Emerald, and Trinity against the dollar spot, and you can see that some doesn't work very well. For example, the Heritage, however, Emerald is still very efficient, or Trinity is still very efficient to control the dollar spot. So this is how we manage to identify if there is in the field or in the lab fungicide resistant strain. Another point that is important for you to know is what we call multiple resistance. So multiple resistance is when you have a pathogen isolate that are resistant to two or more unrelated fungicides. And when we talk about unrelated fungicide, it means that you have two independent mutations that appeared in the pathogen population that are not related to each other. And this is due to the fact that the pathogen has been selected under the exposure of the two chemicals. An example here for the dollar spot, as you can see, we worked on several isolates of dollar spot taken from different locations. And we did the test against boscalid and azoxystrobin. You can see that, for example, here, the 12 isolate that we tested, you have the 12 were resistant to boscalid, but they were also resistant to azoxystrobin. So boscalid is frag number 7, and strobinurin is frag number 11. The azoxystrobin is frag number 11. So here we are in a case of what we call multiple resistance. The pathogen is resistant to two different modes of action, two different groups of fungicide, two different number of frag. And this is due to the fact that the pathogen was exposed to both fungicides during several years. Cross-resistance is when the pathogen isolate are resistant to one fungicide and automatically and simultaneously become resistant to another fungicide from the same class. An example here. So this is a test that was done on 3 different isolates here. This is the control isolate, the isolate under boscalid and the isolate under pentopyrad. These two fungicides are SDHI fungicide and belong to the same frag number 7. And as you can see here, this isolate is sensitive to boscalid, but this isolate is resistant. And the same here, sensitive here and resistant. We are in a case of cross-resistant isolate. The isolate that is resistant to boscalid become also resistant to pentopyrad, even though a pentopyrad was not used in the field. So it's a case where you have the cross-resistant is when you have a pathogen that is resistant to one fungicide and also become resistant to another fungicide from the same mode of action, the same group, the same frag number. If you look at If you want to have more information about the FRAC group, you can look at www.frac.info. You will find all the information you need. The different chemical groups, the different common name, also the target sites of the fungicide, the mode of action of the fungicide, and also the FRAC code. And most importantly, so the FRAC code will will tell you, okay, which one you can mix with which. And also you can have comments about the low or high risk of resistant emergence. For example, here the dicarboxymide, they have the resistance to botrytis is very common. So it has very high risk to have a resistance against botrytis. All right, we know that in Georgia, we have this problem of fungicide resistance. And this is for a lot of fungicide class and also a lot of diseases. How can we manage to reduce this fungicide resistance? The first way, so the resistance management it's important to know that the resistance management will delay but not prevent the development of the resistance. The development of the resistance will always be there. But our goal is basically to delay its emergence at a higher level, like a higher proportion of individuals carrying the resistance in the pathogen. The first way to do it is to always use at-risk fungicide in combination with low-risk fungicide. And this, you can do it in different strategies. Either you can do alternate single application, high risk, low risk, high risk, low risk, or alternate blocks of annulated fungicide, high risk, low risk, dry risk, or in a tank mix, high risk and low risk, mix it together. This is one way to do it. And this is very important to think about it. Another thing is restricting the number of applications or total amount per season. And this is very important. So basically reducing the number of application and basically the amount that you are applying across the season. And one way to do it is to use, there are several disease forecasting systems that are very efficient, that are available, and you can use them to basically know exactly when is the high risk of having a development of a disease. And you can do a preventive application at a particular time when the conditions of the environmental conditions are favorable for the development of the disease. And this will help you to reduce the number of applications. Another thing to know is to maintain manufacturer recommended application rate. The application rate is on the label, and it's very important to keep this application rate at its correct level. Do not try to reduce this application rate, because by reducing the application rate, you may encourage the development of the resistance in your pathogen population. And this is mostly dangerous when we're talking about quantitative resistance. When I say quantitative resistance, I'm talking about the multi-site resistance. We really want to keep this multi-site resistance very efficient because we have problems with the site-specific resistance in the field. Other way to manage the disease resistance, avoid to use eradicant. It's better to apply before the infection than applying when you already have an infection, because most of the time when you see the infection, the population is very, very high of your pathogen, and it's much, much difficult to control the disease. Practice integrated disease management. Use disease-resistant varieties, different cultivars. Try to not use one cultivar, but different cultivars. So there are different management strategies that will help you to reduce this disease resistance in the field. Maintain chemical diversity, use different mode of actions, use less low-risk fungicide with high-risk fungicide etc. So these are very important strategies that you need to keep in mind. Other information, here I give you several websites that are very important for you and will help you to decide and to have a better management strategies concerning fungicide diseases. What are fungicides here? The FRAC fungicide list, pesticide classification system, pesticide database, biopesticide available. This is also very important if you want to have access to biofungicides and not fungicides. And there is a Georgia pesticide handbook that is also very good, where there is a very good information. Thank you. This is what I have for you today. I hope I was not too long.
Thank you, Bochra. This was really great. Lots and lots of information.
I know.
All important. Anyone, we're kind of pushing against 5 o'clock, so a quick couple of questions we can handle. More than that, we'll have to take them at a later time, maybe by email.
Sure. So I put my email here. If anybody has any question, my email is bbahri@uga.edu. I can help you in any, especially if you have a problem with fungicide resistance, do not hesitate to send us samples. We can work on the problem that you have, run it across different fungicides and see what fungicides are not efficient anymore against your population and what we can recommend you to use.
Questions?
I don't know if there is any on the chat.
I don't see anything in the chat or
okay.
in there.
All right.
All right. Thank you, Bochra. Thank you, Allison.
Oh, you're welcome.
Again, this was lots of really important information, lots of chemistry, lots of understanding of biochemistry and plant and organismal relationships, but it's all great information. I'm certain to go back and look at it a couple more times so that I myself can absorb some of it. Thank you all.
Thank you.